Yeah, when there is a heavy rain during the rainy season in Africa, that's when we get a lot of malaria, because there is a lot of mosquitoes, because one mosquito can lay eggs 100 per day. So you can imagine if there are a lot of mosquitoes at the field, the danger is very high.
Yesterday we went to Kimbembe Sub-District Hospital, and the father have 12 were diagnosed of malaria out of 20. You see, it has started coming up again. It used not be here. So I don't know whether because of density or what has happened. Initially, we never used to have such cases of malaria. They had gone down.
Yeah, it is a big burden, because especially where there are irrigation schemes like Kimoya, Ahero, in Kisumu, people around there, they are being attacked by malaria, and they can die when they are young children and young mothers. So they need to be helped.
Since about the year 2000 in Africa, we've observed quite a sharp decline in the cases of malaria. And this has largely been attributed to the increased use of insecticide-treated bed nets. However, in recent years, the number of cases started to flatten out. And this is due to a number of factors, but it suggests that the techniques that are being used, distribution of bed nets and insecticide spraying inside houses, have sort of reached their carrying capacity and are not really having increased benefit. In the last couple of years, we've actually started to observe an increase in the amount of malaria, which is a very disturbing trend. So the microbiome associated with animals and insects can have a very important effect on their health. We know from other research in the field that the microbes associated with mosquitoes can determine whether or not they can transmit the malaria parasite. So with that in mind, we went searching for symbiotic microbes associated with mosquitoes that could be used to prevent mosquitoes from being able to get infected with the malaria parasite. So in a way, we're kind of looking for a vaccine for mosquitoes that protects them from the malaria parasite. Now, some of the key characteristics we're looking for was a microbe which firstly blocks malaria, so the mosquito, when they harbor this microbe, are no longer able to become infected with the malaria parasite. But in addition to that, because mosquitoes have such short lifespans, it's really important that if we manage to infect mosquitoes with this vaccine microbe, that they can then transmit it to all their offspring, so that it stays within the mosquito population generation after generation. So the key characteristics are really a transmission blocking microbe and one that's transmitted from mother to offspring. So we observed the Microsporidian symbiont in a number of different mosquito tissues. Firstly, we observed that it was very prevalent in the mosquito gut, which is actually an important site for malaria transmission, because malaria parasites come in through the gut and have to gain access to the body of the mosquito. Another tissue where we found a lot of Microsporidia was the mosquito ovaries, and we believe that that's how Microsporidia is transmitted from mother mosquitoes to all of their offspring. The Microsporidia gets into the ovaries, colonizes them, and then gets access to the eggs, where it's transmitted to the next generation of mosquitoes. In these microscopy images, you can actually see that the Microsporidia is entering into a developing egg in the mosquito's ovary.
We also decided to screen all our samples for Microsporidia MB, which was prevalent then, for a field correlation with Plasmodium in mosquitoes, which we isolated from Western Kenya in a place called Mbita, which is an endemic region for Plasmodium. And voila, to our surprise, we realized that there was a negative correlation, that each time there was Microsporidia in a host, you couldn't find a co-infection with Plasmodium.
So what we do is, having set up infected lines within the insectary, we get gametocide carriers from around schools in Bita, and from these carriers, we're able to get blood, which we infect our mosquitoes with using membrane feeding assays. So once we infect our mosquitoes, wait for the incubation period of Plasmodium, that's 10 to 12 days, and then we screen these infected lines with quantitative PCR to check for Plasmodium and Microsporidia. And from these results, we are able to determine if Microsporidia has any limiting factor on Plasmodium development within our mosquitoes. So far, we have been able to prove this. We have shown that Microsporidia MB does indeed protect the mosquito from Plasmodium development at both the oocyst level and the sporozoite level.
So we've gotten to a point now where we found this Microsporidian in Anopheles mosquitoes, and we've demonstrated it has some of the really key characteristics for it to be useful for malaria transmission blocking. So mosquitoes that are infected cannot pick up or transmit the malaria parasite, and mother mosquitoes that have this Microsporidian parasite infect all of their offspring with it. So what we need to do now is figure out how we can actually use this in the field to decrease the burden of malaria. So we've set up screen houses where we release infected mosquitoes into a natural population of mosquitoes, and we see how this Microsporidian spreads through those populations. So we understand the parameters, and we understand how many mosquitoes that are infected we would have to release into a population for those mosquitoes to infect the rest of them and result in a population of mosquitoes that's unable to transmit malaria anymore. The next step after that will be to actually release mosquitoes into the field, what we call a field trial. The idea would be to release male mosquitoes, so we wouldn't be putting any of the population at risk. Male mosquitoes do not transmit the malaria parasite, and we would then investigate levels of Microsporidia after releases, and also monitor very closely the levels of malaria. The expectation would be that after a certain number of mosquitoes are released, we would start to see sharp declines in the caseload of malaria over time. And because the beauty of the strategy is its sustainability, because the Microsporidian is transmitted from mother to offspring, these releases would not need to be kept up at the same rate year after year. The Microsporidians would do the work of maintaining themselves and transmitting themselves through the mosquito populations.